Hello, everybody. I'm going to kick us off now. So welcome to this Oxford Centre for Personalised Medicine guest lecture. Before I get started and introduce today's speaker, I just wanted to first briefly introduce the CPM to anybody who's not familiar. We're an outreach initiative based at the University of Oxford, a joint venture between the Wellcome Centre for Human Genetics and St Anne's College. Our aim is to communicate all aspects of personalised medicine with students, scientists, and the general public. And to do this, we host talks, events, we have a blog, and more recently we've been creating short video vlogs and actually animations. You can find recordings of all of our previous talks, including people like Jennifer Doudna, Heidi Ream, and Adam Rutherford, and many others on our website. And please also follow us on Twitter at CPM Oxford to find out more about future events. So on to today's speaker, I'm absolutely delighted to introduce Kumar Veerapen to speak to us about the COVID-19 Host Genetics Initiative, or the HGI. Kumar is a computational scientist at the Broad Institute of MIT and Harvard in Boston, Massachusetts, and he's been central to the scientific communication efforts of the HGI, which is a groundbreaking global collaborative effort to understand the influence of host genetics on COVID-19. If you've got any questions at all for Kumar during the chat, then please put them in the Q&A, and he'll either answer them as he goes through or we'll come to them at the end. So without any more from me, please take it away, Kumar.
Thanks, Nikki. I appreciate the invite to, for this talk today. Just FYI, I'm right now in Malaysia, where it's 1:32 a.m. A little paranoid that I was a little late. So welcome to the talk today. My name is Kumar. I just realized that we're in webinar mode, so the whole microphone stuff is kind of a moot point. So if you want to unmute yourself or mute yourself, it really doesn't matter, or the video. But point is, if you have issues with your Zoom, or if you have questions, please post them in the Q&A, or if you'd like to just post it on the chat. I'm also going to monitor it from time to time. I have two screens, so I should be able to see both of them. So welcome. Today I'll be talking about the COVID-19 Host Genetics Initiative. It's a global initiative, and today's talk will be focused more on how we did some of the mapping of the genetic architecture of COVID-19 while also improving diversity in genetics. As I was mentioning, and as Nikki was introducing me, my name is Kumar Veerapen. I'm a computational scientist with Mark Daly and Ben Neal. So one of the first questions that we asked when we first started the initiative, which was thanks to Andrea Gana, who's currently a PI at FIM, the Finnish Institute for Molecular Medicine, and he worked with Mark and Ben and Rachel Liao, that the first question we asked was, why do some people get really sick with COVID while others don't? I love being interactive during my conversations or my discussions. So this is a poll that I hope that number for the UK should work for everyone here. Otherwise, you can go onto this website, pollev.com/Kumar2022. The question that I'm asking for this poll is, do you think severity and susceptibility to COVID-19 is heritable? So I'm going to give you probably a minute or two just to log into this, and you can answer away, either A for yes, B for no, or C for maybe. So we have one answer so far. So some of you think yes, it is, well, there is some heritability estimate towards it, and some of you think no, which if we think of the classical genetics, when we think about heritability estimates, and I remember this during my PhD, where my mentor was mentioning, and also always learning about it, most infectious diseases have a very low heritability estimate of about 1 or 2%, if not lower. So if we think along those lines of classical heritability estimates in complex diseases, no would be an answer that we think about immediately. However, I think for some of you, or well, most of you at this point, who are saying yes to this answer, it's likely because you have been seeing this variability in susceptibility and severity for COVID-19 that I'm sure the data from the NHS has also shown. A portion of you have also mentioned, maybe about 23% of you have thought that maybe there is a heritability towards COVID-19. implying that some kind of genetics of the host is playing a role towards the infection and also the severity of COVID-19 in terms of the symptoms. So I see that this is still fluctuating, and I see maybe is perhaps increasing in the number of people who are voting towards it. And I see that's kind of stabilized at this point. So based on this question, and even your curiosity right now towards, yes, it is heritable, no, it's possibly not, or maybe, was where we started and continued working towards the host genetics initiative. So most of you think it's yes, and that was really where we started. This slide was adapted from Andrea Gana, where some of the resources in the COVID-19 Host Genetics Initiative includes first the questionnaires and study protocols, which are variable between studies. We do understand that this is a global issue and concern, which is why we approached multiple different countries and populations into providing some of their data towards the meta-analysis that we finally produced. So we also then, upon obtaining the questionnaire data and the study protocols, we had a comprehensive data dictionary, which was curated by Stefano Sherry, and myself and Brooke Wolford from Michigan, now in Norway, have been trying to increase the resources for communication for the lay audiences specifically by using blog posts, FAQs, and our Twitter account, which I'm very excited to share with you. And subsequently, we also do have a catalog of more than 200 studies and research questions, which are again curated, and because there are many cogs and cogwheels towards our study, many people do contribute and lead the different sections of the study that increases how optimal the study has been conducted. One of the major things as geneticists that we're really excited about is the results from the meta-analysis. I would have to remind everyone that today's talk will not be focused a lot on the meta-analysis results, but more on how we communicated and improved our collaborative effort towards forming the global effort of the COVID-19 initiative. And if you do have questions, I do see in the chat, please post your questions for me in the Q&A. And finally, we do also have some in silico follow-up results, which I will not be presenting today just for the interest of time. If you have further questions, they are in our paper, which I will provide a QR code at the end for you to download the paper directly from the Nature website. So one of the things that we also try to address through the initiative is that we do understand a large proportion of genetic data is generated from the European ancestry. One of our colleagues, Alicia Martin, who's currently a PI at MGH, Mass General Hospital, came up with this paper a few years ago, which quite honestly, this plot has been seen in almost every genetics talk that I've seen out there. This plot on the x-axis represents years, and on the y-axis is individuals in GWAS, or genome-wide association study, in terms of millions. As we can see here, and the different colors represent a different population or ancestries globally represented. In terms of studies which have been studying Europeans, which are in red, it's highly disproportionate towards the other ancestries which are available globally. And this on a plot that's just shown right now in the present day of what the global population is like, this is completely discordant towards the global population, which does not see a large proportion of Europeans being represented globally. However, seeing East Asians, South Asians, Africans, Greater Middle Asians, Oceanics, and other populations which are represented in the global population. Considering this disproportionate amount of that have been studied in GWASs towards the global population, this was something that we took while being cognizant towards understanding COVID-19 and obtaining and ascertaining different study populations. Which brings us to this slide over here. We had three different major ascertainment groups. The first would be biobanks, which have really large data sizes, such as the UK Biobank, which most of you would be very comfortable with. Myself, for instance, would be with FinGen because of just the study that I work with, and other sorts of global biobanks that we've seen right now. The clinical studies, which were ascertainment, which was biased towards COVID-19, this to us when we ran our analysis, had the best power towards detecting some of the COVID signals. Some of the things that I would like to point out would be the Biobank Quebec, which had a pretty substantial amount. And I'm trying to look right here for the one from the UK, which I can't seem to find right now. Apparently I'm a little blind towards it. Oh, here, the genetics of mortality in critical care from the UK, which is one of our largest clinical studies that were contributed towards our COVID-19 initiative. We also had some data from direct and consumer companies such as Helix Exome Plus, 23andMe, and 24 Genetics from Estonia. In total, we had about 47 studies from 19 countries. This made up to about 35% of all registered studies, because there were some studies, we had about 143 studies that registered with us, but only 47 had data that was able to contribute towards our entire data set, which comprised of about 50,000 positive cases. And in total, including the non-positive cases, we had about 2.1 million genotype individuals, making it the largest genetic study known to mankind. So moving on, the traits on this study. This is a Lotus Plot, which is something that we came up with at the Broad Institute, just to represent different traits. The different traits that we see here in orange would be the critically ill COVID-19 patients, the reported SARS-CoV-2 infection. This would be both positive in some senses, so reported positive SARS-CoV-2 infection, and finally hospitalized with COVID-19. We did make a distinction between the critically ill COVID-19 patient and the hospitalized with COVID-19 patient, because not all patients that were reported in our data set were hospitalized. So based on this color code is what we'll be observing in the next couple of slides, where the orange would be critically ill, the turquoise or slightly blue, for some of us which are a little colorblind, I apologize, would be reported SARS-CoV-2 infection, and yellow would be hospitalized with COVID-19. The ascertainment was as seen here, where A would be the orange that we observed before, green or turquoise would be C, and B would be hospitalized, where in analysis A for the critically ill patient, those were critically ill but were not hospitalized, we had about 6,000 individuals. For analysis B, those who were COVID positive, critically ill possibly, but definitely hospitalized, we had about 13,000 individuals. And finally, for COVID positive that were not hospitalized, that were, as far as we can tell, not critically ill for COVID-19, just plain positive COVID-19, those that we had about 50,000 individuals. So we analyzed these three data sets separately that I'll show you, well, a couple of them in a few moments. And so the one thing that we did notice was, upon doing all three of the analysis A resulted in the poorest results, and we hypothesized it's possibly because of the very small sample size of about 6,000 individuals. So we only focused for the rest of our analysis on analysis B and analysis C, where B would be hospitalized COVID-19 patients and C would be positive COVID-19 patients. And as I was mentioning and alluding to earlier from Alicia Martin's paper, we did want to increase diversity in studying this global infection. So as we can see on the top left panel, we kind of hit most of the continents globally, not all, but most, which is, well, better than most of the current studies which are out there for genetic studies. So as we can see from the top left panel, panels A, B, C, D, E, and F are as magnified below, where A would be from Western America, from B would be from the East Coast, or well, some parts of the Midwest in the US, and C would be from South America, specifically for Argentina. From D would be from the EU, along with some parts of the Middle East. From E would be, well, the other parts of the Middle East, and on F would be the two countries that we ascertained from Asia, namely South Korea and Japan. Each of the petals that we observe, because as I was mentioning, this is a Lotus plot. The petals, the colors themselves, I've already introduced to you earlier, and the sizes of each petals would represent the effective sample size. The larger the petal, the larger the sample size for a specific analysis type. So if we notice here from panel A, the turquoise colored one is the largest, implying almost 200,000 individuals that were ascertained which were positive. Another thing that we also noticed, because we do know that we had an ascertainment throughout the globe, how about by ethnicity? And when we looked at by ethnicity, we did see a large proportion remain to be European. This was not truly surprising to us, considering that we do know European countries tend to have more resources. Another thing that we also tried to introduce into our ascertainment is to encourage some of these studies to contribute samples. How? One, by only asking for the DNA. Why? Because with our collaboration with Erasmus, we effectively could provide free, if not much cheaper prices for genotyping. This actually improved some of our ascertainment from the Asian countries and also from the South Asian countries. However, we do see, again, a large proportion of which being European. One of our collaborators, specifically the one of mine in Malaysia, could not contribute due to other reasons that we don't really have time to go into, and well, not as exciting considering the data that I'm going to show you in a few moments. So the scientific and public health impact of COVID-19 HTI are as follows. I did mention that it's the largest GWAS ever of more than 2.1 million people analyzed. It's also the largest number of authors on a paper. I think the Higgs boson actually comes close to it of, in our case, more than 3,000 authors. We actually had a script in order to write out the authorship page. It's also containing one of the highest global diversity in genetics known and currently published of about 20 countries. And it's been submitted for publication in order to increase scientific engagement for data sharing and open source. One of the things that, considering this is led by Mark and Ben, is for all of our data to be as open source and as shareable as possible. The one thing that we also encouraged during our entire time from what I think it was May of 2020 till now, is that every meeting of ours will be available on the HGI website. There's a recording, there's the slide decks, there's also any transcripts that were taken during the meeting as well. On top of that, while I led the scientific communications group, we also improved our Twitter presence by making live tweets during these meetings. Finally, we also have all of the data being logged in onto, being saved in the cloud and also available in our HGI website as live links. And we built it truly on existing framework and just continued building with the global partners. And the one thing I would like to also mention is that we use the term partners and not collaborators. Despite Mark, Andrea, and Ben leading the entire initiative, we also wanted everyone to have equal partnership towards the data, equal amounts of responsibility, equal amounts of using the data, or just like amount of using the data. So the one thing I would love to show of how our partnership grew was when we first started analyzing the data and when we had some interesting signals by looking at hospitalized versus COVID-19 negative patients. And this was in, I think, July 2020. Yes, it is in July 2020, where we had about 3,000 total cases versus about almost 900 controls. As you can see here, we only had one peak on chromosome 3, not even the one that was on the MHC marker on chromosome 6. After about 6 months, we increased our fold to about 13,000 cases. And now, instead of only having one significant peak, we now have 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10 significant peaks. And just in case if some of you have never seen these plots before, I apologize. But let me try to just explain it to you just in case. The x-axis is the chromosomes, and the y-axis are their negative log base 10 p-values. Each point that you see here are positions on the genome. The higher the position, the more significant or statistically significant that position is, and everything higher than the horizontal red line is, well, considered extremely significant by genome-wide significance. And so when we had release 5, which is one of our most recent releases, this was led by Juha Karjalainen, one of our colleagues from the Finnish Institute of Molecular Medicine. Again, as I was mentioning, this, well, what I showed previously was known as a Manhattan plot. A reflection of such, this is known as a Miami plot. The minority of geneticists call it a Chicago plot. Really don't know why, because it really looks like a Miami plot. Anyway, probably because I was biased because I did my PhD there. Anyway, moving forward towards what this plot is showing. The Miami plot shows 2 different phenotypes where the x-axis and the y-axis are similar. The x-axis would be the chromosomes, and the y-axis on the top part of the Miami plot would be exactly how you would interpret it as a Manhattan plot. And the lower plot is the lower the point is, the more significant it is. Similarly, the red horizontal line, anything above that threshold, would be known as significant. I don't mean to truly gloss over this, but today's talk is really more about how we engage the community and also try to improve collaborative efforts. But to also show to you that on top of the 10 points or 10 locus of interest that we also had 4 additional locus by looking at individuals who were positive for COVID-19 versus individuals that were not positive, so negative. Each RS number that you see here are also known as SNPs. Again, I'm just trying to gauge everyone just in case if not everyone is a geneticist in the audience. So we had all of these and we did a few different in silico analysis. Some of the analysis that we did are interesting and I would welcome you to read the paper. One of my favorite analysis that I'm not going to show you right now is that we used all of these SNPs and we prioritized them in terms of how significant they were biologically by parsing in a few different kinds of variables into our statistical model to determine which SNP were more interesting than the other. Not just by looking at p-values, but by looking at functional studies which are available. Another thing that I was truly excited about was that as a geneticist, we're always looking at effect sizes and p-values, but what do they really mean? Of course, talking to molecular geneticists, we could probably take some of this and do some knockout, knock-in, cellular, or some kind of model organism studies, but I'm not that kind of geneticist. Instead, this excited me. This is what we call Mendelian randomization. We essentially looked at genetic correlations that were observed between a set of traits versus the SNPs that we obtained. This in concept could give us causal associations, and this concept is then called Mendelian randomization. On the top x-axis are the traits that we looked at. These are just a subset of the traits that we picked out. This was led by Shea Andrews. On the y-axis, the first part are the genetic correlations. The second part are the Mendelian randomizations. The more blue a color is, the lower the correlation or the Mendelian randomization effect. And the more red something is, the higher that effect is or correlation. As we can note here, some of the correlations are somewhat apparent, where the stars are significant correlations in terms of genetic correlations, such as in BMI. The Mendelian randomization just improved some of these signals, and BMI is one of the largest traits that have been shown, or the most frequent traits that's been shown in many COVID-19 papers as an indicator towards someone having a higher or a relatively more severe COVID-19 phenotype. There are a few different results that I would love to show you as well, but due to the interest of time, I would like to move on towards my next section. But before I move on, based on the number of SNPs that we've obtained, and also finally to also mention, other than the Mendelian randomization analysis, most of the SNPs, if not all of them, lead towards lung dysfunction and immune dysregulation. I'm happy to talk offline about some of the signals, but I would like to also gauge, what do you think of the HGI results so far? And this is another poll. I love taking polls, and I blame Heidi Rem for this. So this is an emoticon-based poll. You can go onto pollev.com/Kumar2020, same exact website. I see half of you are happy, the other half of you are, your mind is blown. Just going to take a quick sip of water. All right, so some of you are shocked. If you have any comments on why were you shocked or why your mind was blown, by all means, please place them on the chat. I'd love to hear what your opinions are onto why you were shocked at some of these effects. Why were you shocked at some of the snips that maybe, you know, some of you may have seen them before. And why was your mind blown? I'd love to be interactive with you. So by all means, again, post your comments on the chat or on the Q&A. Love to hear them. Because when I've done some of these talks before, many people their mind was blown because one of the main threads that I've seen so far is, well, one, you don't really see such a large genetic study. And something else to also note that I did not mention is that most of these effect sizes are effect sizes that I've never seen before in a common variant study. These effect sizes are almost close to 2, if not 3.0 in terms of the odds ratio scale. To me, that was mind-blowing. And why we think it's also robust is because these results tend to replicate through data freeze to data freeze. Subsequently, the p-values are pretty strong p-values. And even when we dissect the data further, the odds ratio remain as strong as they were. So our study has shown to be pretty robust, and I really encourage you, if you think the results are exciting enough, please go and read our paper, which can be found using this QR code here. If not, you can access it using this DOI, or if you're lazy like I am, you could just type in mapping the human genetic architecture of COVID-19 and pick the Nature paper. It's the only Nature paper out there. And the part that really excited me is that, you know, it's still the largest GWAS in history of more than 2 million people. I think the GMBI is coming close to it. The Global Biobank Meta-Analysis Initiative that Wei Zhou Wei is actually leading. And it's also the largest number of global collaborators of almost 3,000 scientists. So if you read the paper and hopefully have convinced you a little bit so far, we provide a very high confidence of association between the 9 loci to COVID-19 severity and susceptibility. Looking at severe cases versus population controls is one of the most effective designs, not just seen previously in other genetic epidemiology studies, but also this of ours. Some of our loci do point to clear biology of lung-related and immune-related dysfunctions and therapeutic targets. We do have groups which are looking towards doing some of these future in silico analysis in order to guide the therapeutic targets. So please be on the lookout for more work coming out from our initiative. And we're working together as an open community, which allowed us for robust real-time replication of findings and exchanging of results between studies. And this actually brings us to a segue towards my next slide here. We brought together minds from all over the globe to address this pressing global concern. We know that COVID-19 is we're still out of the woods yet. And when we work throughout it, and we're still working towards improving a lot of our analytical results, one of the main things that we used was Slack, which Nikki and mine, one of our colleagues, Konrad Krzyzewski, actually reminded us that Slack is actually a short form for searchable log of all communication and knowledge, which I think is pretty cool because when I first thought of Slack, I just thought I was slacking off at work. Oh, well. So I would like to also show you this plot over here. So this is just something, if you are aware of Slack, it's like an IM or instant messenger, like if some of you are using Microsoft Teams or you know, I can't even think of other methods of communicating with your colleagues other than Slack and Teams. But if you have others, well. So it's very similar to that. And one thing that some of us are interested in, especially in the analytics, is how frequently were we using Slack? So this point here, I just wanted to show that on the week of December 6, 2020, which was one of our biggest weeks before the release of the plot that I was showing you earlier from January of 2021, we had the highest number of active members of about 624 individuals and people who kept posting. This also included some of the people that were just personal messaging each other, showing that this was pretty effective at that point. And, you know, it was variable throughout with still pretty significant number of people that are using Slack to communicate with other scientists over the world. And so this brings me towards like how effective the COVID-19 HGI was, because we had on average about 240 to about 260 people weekly using Slack, just using the COVID-19 HGI Slack. And this improved our goal towards increasing the diversity of how we continue using the data to answering other forms of COVID-19 questions. And some of the questions here are related to, well, most all of them are related to COVID-19, but what I wanted to mention was they are all listed on our website under the covid19hg.org/projects. My team and I actually came up with, like, we tried our best to make the website as user-friendly as possible. So if you have comments upon going through the website, please feel free to send me an email or just leave us a comment on our Twitter account. We're happy to take care of what you think could make our website look better. So going through some of the projects that we have and active projects: complication of blood vessels, lung-related distress, because we know many of the SNPs and loci that we obtained were lung-related diseases, clinical implications from chromosome 3, and when it's italicized and bolded like this, it's when the groups have the publication. I think right now it's already in full print and not in preprint. Blood cell development and complications, because we do have a huge group just looking at histo. And so these, the blood vessels and blood cell group are actually from the same groups. And when I mention same group, it's not from the same center or same country, but instead it's just multiple people from multiple countries and multiple institutes in one Slack channel discussing that topic. Statistical causality of traits and genes. This is actually led by Shay Andrews, the Mendelian randomization slide that I was mentioning I was excited about. So they're in the process of publishing that particular work. The whole exome sequencing and whole genome sequencing analysis, which I'm a part of as well, looking at sequencing data instead of just genotyping data. All of the data that we have right now, all the data that I've presented to you, are all using genotyping data. We're moving towards the sequencing data, and one of the things that we've been trying to do is to ensure that all the sequencing data is analyzed as, what's the word that I'm thinking of, a standard across all of the studies before they provide us the summary statistics for analysis. And that's one of the things that I've been doing in the background of advising some of our collaborators on what to do and what not to do, especially using the tool that I give support for called HAIL. And finally, the long COVID group. We do know that a non-zero proportion of the population which have been infected by COVID have had long COVID symptoms, meaning that even though they've been infected and after the 2 weeks of the infectious period, they continue suffering from COVID symptoms like no taste, no smell, sore throat, etc. And we have a whole group looking at just the host genetics of understanding the long COVID syndrome. And so we have all of this work, all of this cool science, but how do we communicate our results to a community of scientists in a world that's eager for information? So myself and Brooke Walford, previously of University of Michigan, one of the things that we've been keeping in mind by communicating most of these things is we want a culture that is inclusive of everyone and where everyone who joins feels that they have opportunities to succeed and grow. And that's really how we also led our group. As I was mentioning earlier, everyone that's part of our study, they're not collaborators, they are all partners in the group, which is why we're trying to give opportunities to as many people to use the data to empower their own study or their own scientific curiosities. In our team specifically, which is a scientific communication team, we have multiple subgroups as well. In each subgroup, we've given them as much legroom in order to grow and to build up on things that they're interested in communicating with the world. Before I move on to some of the efforts that we've done, one more poll. I swear it's one of the last polls. I love interacting with people, and I know I can't hear your voices, but this is the best way for me to hear your voice. How do you, the audience right now, obtain information from a field that is not in your expertise? This is a donut plot. I know it looks like a pie chart, but because there's a hole in the middle, apparently it's a donut. So answer accordingly, where A would be blog posts, B would be reputable websites, C would be, well, I see some of you are already learning how to use PolEV. This is perfect. And you know, it could be others. And if you have any notes on what else you've actually used, please feel free to let me know in the chat as well. I see that we have like an even split. Well, not so even right now, but most of us are in reputable websites. We do have a large proportion right now in social media, which sometimes is a good thing and sometimes it's a bad thing. And what I've been learning upon being back home in Malaysia for the past 2 months is that social media not exactly a great thing when it comes to like, my older family members. If you have that issue as well, more power to you. Many people also look at reputable websites, and I love the proportion that I'm seeing in my audience right now, because most of the audiences that I've spoken to before are Americans, and they seem to love going onto blog posts for their information that's not in their field. So this could be like, for myself as a geneticist, not very comfortable in, what the Higgs boson is. Still really don't exactly know what a Higgs boson is, but how I read about it was by using reputable websites, news articles, and sometimes talking to my gal, guy, and person pals. So I see most of you use reputable websites. A large proportion of you use social media, such as Facebook and Twitter. If you have specific social media that you use, I'd love to hear it in the chat. I see that some of you use blog posts and news articles. This is all really cool data, and I can't wait to just look through it in detail later. So thank you for your contribution. And based on that, I hope that our website is a reputable website for you, because we do have blog posts on our website. And one of the reasons we have this blog post is to allow accessibility to our scientific results to everyone and not just the scientists in the room. We do actually have 2 prongs in terms of our blog post that we're publishing, where the first prong that I'm showing right here is for the layperson. We do have a separate prong, which is just for the scientists who are, not mainly geneticists, but just people who understand a little bit more science. And we cater most of our blog posts to the layperson. And it's something that, just in case that you've heard me say a few times at this point, data freeze. So a data freeze, what does it mean? We freeze our data every 1 to 3 months. And once we declare freeze, we run our analysis. After we reveal the results, we accept more data, add it onto the freeze that we previously had, and we repeat. And so every freeze that we have, where the first freeze, we published a blog post, was in July 2020 that I co-authored with Brooke. This was our first foray into writing a blog post. And my late mother tried to wrap her head around this. And she was like, how do I try to understand what you're writing? She was one of my biggest fans of my writing. And so she was like, this is not easy for me. This is a little harder. This is easier. And she was a nurse during her entire life. So she also gave me a lot of input, and Brooke's parents, Brooke's family also gave her a lot of input. So we do have someone who had a question on what is the big peak at chromosome 3. It's actually a whole region that we have on the OAS genes, and we have a whole paper that's coming out. And if you need the link, please feel free to send me an email. I'll be happy to pull up the preprint that we have on how we fine mapped towards chromosome 3. I hope that answered your question. So returning back to the blog post here. So something else that we also noticed was that a very large proportion of the world does not speak English. So we just asked out to all of our scientist friends in the HTI on what languages would you like to translate. To my surprise, almost everyone wanted to contribute towards our translation efforts. And some of these efforts are as listed here. And of course, Bahasa Melayu, which is the language that we speak in Malaysia, is highlighted because I kept clicking on it. But to note, the translations to multiple global languages improves inclusivity and accessibility to science, which is one of the major things in science that, as scientists, we don't really realize. We just talk to each other, but we keep forgetting that, in non-English speaking countries, they don't learn science in English. So one of the things that I loved about this effort was how easy it was to work with all the people. I had about 100 translators. Oh, sorry. Yeah, I had about 85 translators that translated to about 30 different languages throughout the globe. And the part that I love the most is that all of these are scientists, and some of them really big names in the field, and they still took time out of their busy lives in order to translate our blog posts. Why? because they wanted to increase accessibility of science. So TLDR, too long, you don't read, it's fine. We had about 30 languages throughout most of our freezes. Another thing that we also noticed was that throughout our blog posts, from the multiple iterations that we had, people actually had more questions about what genetics was. And also a few things about what a host genetics versus viral genetics was. So we also improved efforts into increasing genetic literacy, where right now, James Priest, as shown here, led the effort onto coming up with our FAQ page. And so one of the first questions we usually ask is, I mean, as a layperson, I have the genetic variants. I have one of the 10, maybe, maybe you've done your own genetic screen and you happen to see that RSID. And if you have a variant, does it mean you're more likely to get sick or have a severe outcome? To some of us, it may seem simple enough, especially for a geneticist. It's all based on probability and likelihood functions, but not everyone can think that way. Another major mode of communication that we used was Twitter. I did notice a large proportion of you use social media for your information in something that you possibly didn't know. Why did we use Twitter? So as to simplify it further, that 142 characters allows you to simplify very extensive amounts of science into very small amounts of tweets, and it's really quick to read. Nikki Whiffen is one person who does tweet pretty often and pretty well as well. So inspired by such Twitter accounts, we then used Twitter as our main mode of communication, and we've been using Twitter a lot. And it's truly because it's a global collaboration of over 24 different time zones. Slack was cutting it, but it wasn't truly cutting it into getting it to people who are not part of the HTI. So if you'd like to follow us, this is our Twitter handle at COVID-19_HTI. By all means, please add us right now. We're still pretty active. And I would like to share with you some of the engagement that we've done, because, we know Twitter works, but how well does it work? So when we posted our major release in January 2021, this was our biggest release and possibly our most significant release, because even our current release just essentially improves our signal. But this release in itself was our best. We had about 19,000 impressions and about 349 engagements. So people who have, retweeted it or did, sorry, I just wanted to check the chat, or had a stream or a thread that they added onto it. This improved significantly when we had our preprint that was based off of this release. This preprint was published in medRxiv, and we had about 71,000 impressions with about 72 media views and about 2,000 total engagements, which was huge. And this is probably my American humor, where through all of this, I should celebrate through interpretive dance. All of this was really led by Nicole Duker with some help from Brooke and myself. And one thing that we did do was we had a DNA Day event last year in April. The announcement tweet in itself had more impressions than our blog post release, which to us was a little shocking, but kind of encouraging. And we were happy to also note that we are one of my favorite tweets right here, which was our COVID-19 Host Genetics Initiative, wishing everyone a happy DNA Day in multiple languages. And this is one of my favorite GIFs in this. We didn't have crazy amounts of retweets or likes, but it was one of my favorite tweets. And a little bit cool for me is that, considering I grew up in a multicultural country, I contributed towards 5 different languages on this. And we just asked most of our translators to contribute towards this GIF. And it was one of my favorite things of our tweets. And so we then also pulled all of our tweets into an Excel spreadsheet just to continue analyzing what we did. During our DNA engagement event, we gained about 64 followers, which I know doesn't sound like a lot, but we gained a fair amount during our previous tweets of our releases and our preprint. So when we had our announcement, I did mention we had about 20,000 impressions. And we did see, highlighted in yellow, some of our highest, most impressionable tweets. What we also did was throughout the day made focus points on, or focus tweet threads on our different main people like Gita Patak, Masa Kanai, who are our two major in silico analysis people, Mari Niemi, who led most of the writing, and Andrea Gana, who is our person, our PIC, our PI in charge. And so we had also a significant number of media views as well. And we did have a media bootleg, was it called a bootleg camp? I actually forgot what camp it was, but essentially we invited a bunch of people, media from different parts of the US and parts of the globe to come to the Broad in order to listen to some of our spiel about how the COVID-19 HGI really took off. And so we really tried to effectively communicate it and use social media at the Broad retreat as well last year. Finally, one of our last things that we did was podcasting. And in podcasts, I do see a question from Chantal, which I'll take after this slide, if you do not mind. So podcasts allow for multitasking and increasing COVID-19 engagement, which was led by Nathan Pearson. We had a podcast in plenary session that had about 6,100 listens, and on Naked Geneticists, which is a UK general audience, so you all would be able to check this out. And this is one, according to what we were told, one of the most popular podcasts that they have that Nathan was on. We also have some ongoing one with Colombia and also translating the audio from our blog post to multiple different languages to increase accessibility of our science. So Chantal Bhakti Villiers said, thank you. What would be the largest impact of the GWAS findings on public health and patient care? So she also has a separate question, but let me answer the first one first. So I would think the largest impact would be, well, one, GWAS can be used in order to detect robust signals. You just apparently need large enough sample sizes. Some of these variants and locus of interest could be used further upon interrogating by, looking at Mendelian randomization method and other in silico methods that we're applying right now to see how robust the signals are. These signals are not truly interesting enough for patient care yet. Again, yet with an underlined bold italicized, because we're still ongoing in some of the analysis. As we know, a lot of the GWAS findings, even non-COVID science related, are not as effective into predicting healthcare and patient care symptoms. So a separate question that Chantal had was, how can these types of studies be used for other infectious diseases when they don't have GWASs in millions? I'm so sorry, but I don't think I have an answer for that, because as a statistical geneticist, I definitely would say the millions would be helpful. Another thing that this could also be helpful towards guiding future study designs is we do realize that the severe versus the totally negatives seems to have the highest signals because of just like how the odds ratio function works. So what you can see as a proof of principle in the COVID-19 Host Genetics Initiative is to use it as future study design methods. So that's really what I think could be used in other infectious diseases. I hope I answered your question, Chantal. Thank you. So to end, what's next? Well, we are further still improving diversity in genetics. And this is one way that I believe we could further do it, because many people are concerned about COVID and many people are studying COVID. So eventually we would have, a further iteration where we just had our data freeze. We actually just released parts of our data last month, but sadly I wasn't there at the meeting due to personal reasons, which is why I'm not as comfortable in showing you that data until I've actually gone through it myself. And finally, one of the things that I'm most excited about is once we understand more about COVID, hopefully we can all crowd at a beach again and get a tan. So I'd like to really acknowledge everyone here of all 925 affiliations of about 47 studies of about 19 countries, led by Mark, Andrea, Rachel, and Ben. This is one of my favorite slides that one of my SciComm members from actually I completely forgot where in Canada, that did this for me. I wanted like a credit slide of everyone included, all 3,300 people who are on our author list, that I would really like to thank, because no person on this list did any less or any more than the other person. Everyone is a partner in this global partnership. If you have any questions, by all means, please let me know via the chat, via the Q&A.
Thank you very much, Kumar. That was great. I've got a question actually. So what do you think are the major barriers to us having kind of truly diverse genetic studies? And what do you think are the key steps to the key barriers that we need to break down?
I think resources is one of the major barriers. And, you know, being like growing up scientifically here in Malaysia, I see those barriers. Funding is a major issue. Understanding of the importance of certain studies like discovery methods in genetics, those are two really major barriers. And, you know, the resources could be, I guess it could be dissipated or diffused into how the political climate is in certain countries, which, you know, even as we see with vaccine efficacy, we do observe the countries that do not have as much resources into obtaining vaccines having higher amounts of COVID, higher prevalence of COVID-19. Did that answer your question, I hope, a little?
Yeah, there's a great one from Alexandra in the chat. What top piece of advice would you give to somebody just starting out in SciComm?
I truly wish I'm still leading this effort, because I would say, Alexandra, come and join me. Always looking for people to write blog posts and to manage some of the other things, because we really had a lot going on at one point. So I would really just say start on Twitter first, start following people that you find interesting scientifically and see how they tweet and see how they communicate. Next thing, also decide on what audience you want to do SciComm for, whether it's for the scientific community or whether it's for the layperson. And that will actually kind of drive you towards what you feel like you want to do. I'm by no means an expert at SciComm. I learned literally on the fly. And this is just things that I've learned, like picked up from people, like, you know, reaching out to people on Twitter and saying, I need advice on how to write a blog post. I need advice on what to do here. I'm trying to think of what's his name. It's like 2:30 in the morning and I can't think of what's his name. But the person who's really famous that writes in the New York Times all the scientific stuff. Nikki, do you remember his name by any chance?
I don't know, I'm sorry.
It's okay. So, I mean, point is, we reached out to people in order to ask, how do I improve on some of my skills here and there? And that's really what I did. And I think it was helpful for me because I had that framework available for me. The number of people who were excited, having more people, and the website in itself for me to just show all of the work that we've done. But if starting out from scratch, I would reach out to someone that you know is good at it, continuing still doing this work and asking for more advice. The more advice you have, you know, the easier things become, just like how science is. Chantal said, thank you. Yes, I hope that we can get a better change, a change in science. Alexandra, I hope that answered your question. If it didn't, by all means, please send me an email here or just like follow me on Twitter and just send me like a personal message. And I'm happy to like chat further offline with you where I can actually see your face and talk to you. But yeah. Any other questions, anyone? Thoughts?
Perfect. I can't see any more in the chat, so I think that just leaves me to say a huge thank you for staying up so late or so early, Kumar, and talking to us. And yeah, I'm gonna say go and get some sleep.
Thank you. Oh wait,
Right, thank you.